Okay. Today we are going to cover the cytoplasm. So it is the part of the cell present outside the nucleus and inside the cell membrane, right? So here we have the cell. This is cell membrane. Here we have the nucleus. So cytoplasm is present between the cell membrane and the nucleus, right? Here.
Yes.
Yes. Okay. So that fluid material is called as cytoplasm. It has the various organelles to perform various activities or function, okay, of the cell. So what are the cell organelles we have? We have nucleus is there, Golgi apparatus is there, endoplasmic reticulum, lysosome, cytoplasm, chloroplast, cytoplasm,
Sir, we can't
Sir,
hear
yes,
you.
sir. Yes, sir.
I was disconnected. Are you there, everyone?
Yes, sir. What just
Yes,
happened?
sir.
I think I was disconnected. I was like explaining and I was waiting for your response. So I didn't got your response. That's why I checked. So it was reconnecting. Okay. So what we discussed here, cytoplasm part. Where were we? I think cell organelles, no? That one?
Sir, yes, sir.
Okay. So organelles are primitive in prokaryotic cell. Primitive means not well developed. Okay. So like we already gone through eukaryotic and prokaryotic cell. So in prokaryotic cell, like cell organelles were not well membrane bound. So they are primitive in nature, right?
Yes.
Yes. Okay. Now we have knowledge enhancer. Very important. What we have on the basis of membranous converting, all cell organelles are divided into three types, beta. Okay, like their envelope is there, which is like single membrane or double membrane bound. Okay, like this. So membrane less organelles we have ribosomes. They do not have the well developed membrane. Okay, single membrane we have. Do remember, without membranous we have ribosomes. Single membranous we have endoplasmic reticulum, Golgi bodies, and lysosomes. Even vacuoles are there. Double membrane bound organelles we have mitochondria and plastid. Plastid are present only on an plant cell, not on animal cell beta. Okay. Okay. Yes. Okay. Next, cell organelles found in eukaryotic cell. Why? Because they are definite.
Sir, can
Okay.
you go up once? I just want to take a screenshot.
Okay. Let me do once done. Everyone. Done.
Yes, sir.
Okay. Now, different cell organelles we are going to like understand one by one. So whatever the cell organelles we are going to see here, they are of eukaryotic cell beta. Why? Because only in eukaryotic cell organelles are definite. That's why we can observe them separately. That's why we are studying here organelles of eukaryotic cell. Okay.
So,
Okay. So first we have endoplasmic reticulum. In short, it is represented by ER. So we have two types of ER beta. See, can you see that dotted structure on ER?
Sir, we
Right?
have smooth ear and rough ear.
Yes. So that if ribosomes are attached, so they are called as
Raffia,
RER, rough endoplasmic reticulum.
particular.
Okay. Yes. If ribosomes are not there, so they are called as smooth
Smooth.
endoplasmic reticulum. Okay, everyone?
Yes, sir.
Okay. So this is the structure. Here we have like ribosomes and they are the tubules. Okay. Number one, it is first observed by Garnier on 1897. When? 1897. Then endoplasmic reticulum term was given by, okay, the term endoplasmic reticulum was given by Porter on 1952. Okay, the membranous network enclosing the fluid filled lumen present throughout the cytoplasm. Okay, so it is present throughout the cytoplasm from nucleus to the cell membrane. It is absent in prokaryotic cell. Why? It is not well membrane bound in prokaryotic cells. So it is not absent, we can say. It is made up of three type of elements: cisternae, vesicles, and tubules. Okay, they are cisternae, they are vesicles, and they are tubules. Okay, on the basis of presence or absence of ribosomes on endoplasmic reticulum, we have two types: smooth and rough endoplasmic reticulum. Easy.
Yes, sir.
Okay, now we are going to understand functions of endoplasmic reticulum. So endoplasmic reticulum help in transportation of material between various regions of the cytoplasm or between the cytoplasm and the nucleus. So it helps in transportation of material. It acts as a cytoskeleton and provide the mechanical support and shape to the cell. Okay, so it provides shape to the cell. So it is responsible for the structure and shape. That's why it is called as cytoskeleton of the cell. Rough endoplasmic reticulum helps in synthesis of protein because ribosomes are attached there. And ribosomes are the functional unit of protein synthesis. Like DNA is also there, mRNA is also there, but ribosomes are also there, which are sent to the various places in the cell depending on the need. Okay, these proteins are being synthesized, and as per the requirement, these are being transported to the various parts. Okay, smooth endoplasmic reticulum helps in synthesis of lipid. Lipid means protein plus not protein, fat plus alcohol becomes lipid beta, lipid. Understood? So they help in formation of lipid. Some proteins and lipid synthesized by endoplasmic reticulum are used for producing cell membrane in the process called membrane biogenesis. Okay, so this fat and protein or lipid or protein which are used for the formation of cell membrane, beta, cell membrane. Okay, and it is called as membrane biogenesis. Membrane biogenesis. Okay, and that protein which is responsible for formation of cell membrane is called as glycoprotein. Glycoprotein. And lipid which help in formation of cell membrane is called as glycolipid. Understood, everyone?
Yes, sir.
Okay, take a screenshot. Some proteins and lipids synthesized endoplasmic reticulum also works as a hormone and enzyme. Okay, so like proteins synthesized by them, they act as a hormone as well as enzyme. Endoplasmic reticulum, okay, form cytoplasmic framework and provide surface for the biochemical activities. Whatever the chemical activities take place in our body, endoplasmic reticulum provides the surface area. Okay. In liver cell of vertebrate, smooth endoplasmic reticulum helps in detoxifying many poisonous and substance and drugs. Okay, so it is the work of smooth endoplasmic reticulum of liver. Understood?
Yes.
Okay. Take a screenshot then.
Take it.
Everyone done?
Yes, sir.
Aman, Harini, Chaitanya. Okay. Okay, so I want you to repeat that beta once. Function and structure. Done everyone? Good plan.
Yes, sir.
Okay. Now we have Golgi operators. Okay. So the structure of Golgi operators is like this, right in front of your screen.
Sir.
Yes.
So once, can you go up just once? Okay, sir.
Okay. So now we're going to discuss about the Golgi operators. Okay, this is the structure of Golgi operators. Okay, it is first described by Camille Golgi. That's why on his name it is named as Golgi apparatus or Golgi body in 1998 by using the black reaction staining technique. Okay, the black reaction staining technique by which it is appeared in the cell. In animal cell, it is live above the nucleus, and in plant cell, it is scattered in the cytoplasm and is called as dictyosome. Okay, so it's scattered form everywhere. So it is called as dictyosome in plants. But in animal cell, it is called as Golgi and it is just present above the nucleus. Shape varies and depends on the functional state of the cell, okay, called pleomorphia organelles. Okay, generally its name, like work is to convert or the modification of the protein into enzymes and forms the, what? Like vacuoles and lysosomes packaging material. It consists of the set of smooth, flattened cisternae, even vacuoles and vesicles also. Its membrane is connected with the membrane of endoplasmic reticulum and make another cellular membrane system. Okay, so it is again another cellular membrane system, and its main function is the modification of protein into enzymes. Now, function. The Golgi operator helps in packaging. See, it is packaging, dispatching various material synthesized by endoplasmic reticulum to inside and outside the cell. It helps in storage, modification, and packaging of the product in vesicles. So, modification also. Sometimes it may form the complex sugar from the simple sugar. So, it forms the complex sugar also from simple sugar. It also forms the lysosomes by converting the protein into enzyme. See, it converts the protein into enzyme and packed inside the body is called lysosomes. Okay. So, do you know Golgi body is a single complex in animal cell, while in plant cell it is formed the separate unit called dictyosome. Okay, we have already gone through that. Is it everyone?
I don't think.
I'm
not sure.
No, here we gone through here. Where? Over here.
Oh, okay. Yes.
Okay, repeat it once, then we'll move further. Done.
Yes.
Everyone. Okay. Now we have lysosome. Again, very important. So lysosomes appear like this. Okay. Okay. So this is the lysosome. So it is discovered by D. in 1955. It is the membrane-bound vesicles formed by smooth endoplasmic reticulum or maybe by Golgi apparatus. It contains a strong hydrolytic enzyme capable of digesting and breaking down the organic material or waste, okay, from of the cell. These enzymes are synthesized by rough endoplasmic reticulum. Okay, so it's like become very important because it is used to digest, the waste material of the cell. So it is full with enzyme, digestive enzyme. Now, function. Lysosomes help in intracellular digestion. See, intracellular digestion, so called as digestive bag. It is also called as digestive bag. It destroys any foreign material inside the cell, such as bacteria. So it used to destroy the bacteria also. Also remove the worn out and poorly working cell organelles by digesting them. So if cell organelles are not working and they become non-functional, okay, so lysosome used to digest them. Hence, make a waste disposal system of the cell and keep the cell clean. During the disturbance in the cellular side metabolism, lysosome get burst and its enzyme digest their own cell beta. That's why lysosome is also called as suicidal bag because it digests or on its own. That's why it is also called as suicidal bag. Understood? Okay. Then we have mitochondria. It becomes very important because it provides the energy to the cell. That's why it is also called as powerhouse of the cell. Okay. So it was first observed by Alumni and was later explained by the Colliker. So name mitochondria was given by Benda on 1897-98. The rod-shaped double membranous cell organ is called powerhouse of the cell because it provides energy in the form of ATP from the carbohydrate. The outer membrane is very smooth and porous, but inner membrane is very folded into the finger-like cristae and ATP generating reactions taking place there. Okay, so they are nothing but, this is nothing but the structure of mitochondria. Okay, now on cristae, oxysomes are present, which are main source of ATP molecule. ATP is adenosine triphosphate. Adenosine triphosphate. So function. So main cell organ is that helps in aerobic respiration. So it helps in oxidation of carbohydrate, okay, into the carbon dioxide and water molecule and releases energy in lot, like more amount. That's why it is called as powerhouse of the cell also. So it performs aerobic respiration there. It releases energy required in the form of ATP called adenosine triphosphate, and ATP is called as energy currency. Okay, it is also called as energy currency of the cell. Okay, everyone. Okay, okay. ATP is used for making new chemical compounds and the mechanical work better. So ATP generally is energy rich molecule and which is used for the different purposes of the cell essential. Okay. So energy rich molecule we have ATP and energy is stored in the form of phosphate bond in ATP beta. So ATP is energy currency of the cell, you can say. Mitochondria have their own circular DNA and ribosome, so regarded as the semi-autonomous and self-replicating cell organelles. So it is called as self-replicating cell organelles. Okay.
Okay.
Yes. So just revise once. Mitochondria is very important. That's why revise once. Okay, done everyone?
Yes, sir.
Okay. Now we have plastid. So it is a cell organism present only in plant cell. Okay, not in animals. So it is the like part where photosynthesis reaction takes place. Okay, so here where it is the outer membrane, here we have inner membrane, that one. Okay, then here we have granum. Okay, here we have thylakoid. Here this material is called as stroma. Here they are called as granum. Okay, grana is, you can say grana also. And here we have like vesicle like structure called lamellae. Okay, they are called as what? Lamellae. Okay, so this is the site of photosynthesis beta. So the term plastid was given by Haeckel on 1866. Okay, it is found in all the plant cell and some protista also. Protist means unicellular organism. They are eukaryotic in nature. They do contain protist like fellow like what chloroplast you can say plastid. There are this is double membranous structure and have their own DNA. Deoxyribose nucleic acid is present there. Thus, it is called as semi-autonomous and self-replicating cell organisms. Self-replicating cell organisms. Okay, because it do perform the replication of DNA. That's why it is called as self-replicating organisms. In photosynthetic prokaryotic bacteria, okay, like cyanobacteria, you can say they are called as blue-green algae also. They are associated with the membranous vesicle, but not with the plastic. So they do contain like membranous vesicle, but plastic is not there. But chloroplast plant is present their part. That's why they used to perform photosynthesis also. Okay. Schimper used the term plastid for the first time in 1885, okay, and classified plastid as leucoplast, chromoplast, and chloroplast. So chloroplast is green, leucoplast is like colorless, okay, and it is responsible for like plastid that stores starch, oil, and protein in it. Chromoplast is like colored plastid, and green in color, okay, contain more color than green beta, like red, yellow, orange, with several colors. Contains several pigment and which provide color to the petals of the flower. Fruits during the ripening, it provides the color, right? Because when mango ripes, it becomes yellow, orange, red in color, right? It is due to the chromoplast. Now, chloroplast, the green plastid found in leaf and green parts of the plant. It helps in photosynthesis. That's why it is called as kitchen of the cell, because whatever the food is formed is in due to the chloroplast only. In chloroplast, that's why it is called as kitchen of the cell. Two membrane of the chloroplast surrounded the colorless proteinous matrix called stroma. It is a colorless protein like matrix present inside the plastid. It is called as stroma, which contain the DNA, ribosomes is there, even certain enzymes are there which help in biological process take place in the cell. The stroma, dark reaction of the photosynthesis takes place here beta on stroma. Okay, dark reaction takes place. Dark reaction means the conversion of chemical reaction, okay, into the food. Here there is no need of light. That's why it is called as dark reaction. I hope you understood. Stroma
Yes.
contain flat, membranous, and disc-like structure called thylakoid. So disc-like structure are present there, as we already gone through in the picture. And their group forms the granum, and their group from the granum. So here single is called as thylakoid, and group is called as granum. So grana are connected by the stroma lamella, and they are connected with each other, right? Like this, by the lamella or stroma lamella. Inside the grana, chlorophyll molecule are present. So in grana, light reaction means photosynthesis takes place. Okay, light reaction means conversion of light into chemical energy. Okay, the grana is the site of light reaction during the photosynthesis, while stroma is the site of dark reaction. Okay, everyone.
Okay.
Then we have ribosomes. OMG. It's quite very big part and detailed one, very good. Now we have ribosomes. Ribosomes were first observed by the Claude in 1941 through the electron microscope and are called as microsomes also. The name ribosomes was given by the Palladay in 1955. The smallest cell organelles found in both prokaryotic and eukaryotic cells
Sir, I
are.
have a doubt.
Yes, madam.
Faraday here is Michael Faraday, right?
No.
Scientist?
Palladay, Palladay, Palladay.
Okay.
Yeah. Michael Faraday is from physics. He has given electromagnetic induction. Right? Okay. Palladay. And ribosome is the smallest unit of the organelle of the cell, you can say. And there is no membrane is present around it. So it is are of two types on the basis of size. Okay. So and Swedberg unit or sedimentation coefficient, like we have 70s and 80s ribosome. Okay. They are made up of two subunit beta. One is called large unit and one is called small unit. So large unit in 70s chromosome, okay, we have like 50s, 70s, etc. And in 80s ribosome, okay, the units may be like one is 60s, another is 40s, like that. And they help in protein synthesis beta. They help in protein synthesis. This is the only function of the ribosomes. Next, we have centrosomes. They are formed by granules called centrioles. Beta. They are formed by centrioles surrounded by the transparent area called centrosphere. Centrosphere. And they help during the cell division. Beta. Okay. They are found in animal cell. Okay. And not on plant cell. Beta. But in animal cell, in RBC and nerve cells, there is no place for centrosome. They are also found in most of the protista, like mitotic plant cell. Sorry, motile plant cell. Motile means if they are long cylindrical cells, so they are called as motile. They are like their centrosomes are present. Function. They form the esters during the cell division. Okay, so they are like splits during the cell division into two parts. One part reaches to one pole and another reaches to another pole. And they produce the spindle fibers, and these spindle fibers help in nuclear division. Beta. They help to split the chromosomes. Okay. Okay, they form the basal bodies of the cilia or flagella. Okay, so like cilia or flagella, they are nothing but the organ which are present on the outer side of the cell, and they help in locomotion of the unicellular organism. So there they are present. Yes. Then we have peroxisomes. They are the small membrane bound sac contain powerful oxidative enzyme, mostly found in kidney and liver cell. Beta. They are found in kidney and liver cell of animals. In plant, it helps in photophosphorylation. Okay, so it helps in photophosphorylation and carrying out some oxidative. See, they are filled with oxidative material, so they help in oxidative reaction and helps in removal of the toxic substance from the body. Last but not the least, we have vacuoles. Okay, so these are unimembranous sac stored in the solid or liquid nutrient, and outer membrane, okay, of it is called as tonoplast. Tonoplast. The outer membrane of vacuole is called beta tonoplast. The small and few animals cells, okay, but large and wall develop in the plant cell only. Okay, they are few in animal cell but large in animal cell. Oh, sorry, plant cell. centrally placed in the plant cell and may occupy 50 to 90% of the cell volume. So in animal cells, they are present in large number, but they are smaller in size. But in plant cell, it is present in a large like size like this. Here we are nucleus. So this is the vacuole, very large in size. Main function is to store the protein, amino acid, sugar, and various organic acid also that are essential for the plant growth. In amoeba, stored food called and called food vacuole. So it used to store the food in plant amoeba cell beta. So it is called as food vacuole. In some unicellular aquatic organisms, vacuoles help in expanding the excess of water and some waste from the cell and called as contractile vacuole. Okay, so these are on the basis of their function. We have different names to the what vacuole. Then we have cell inclusion. So on the basis of nature, cell inclusions are classified as reserve product, okay, like carbohydrate, fat, oil, yolk in animal cell, protein, etc. They used to reserve that. Secretory product, we have enzymes, pigment, nectar in plant, okay. They used to store the waste product in plant, minerals, crystals, gums and resin, latex, even thylakoid also, nicotine, morphine and quinine, tannin and essential oils. So they are nothing but others. They used to help in spread of this material. Okay. So this is all about all the cell organelles. So I want just repeat everything once beta and then we'll continue further. Just repeat it everyone. Everything.